We know, and we've known for 100 years now, that all cancers are due to abnormalities in DNA. Abnormalities in DNA which are present in the cancer, but not in any of the normal cells of the body. Today, for the first time, in two individual cancers, a melanoma and a lung cancer, we have provided the complete list of abnormalities in DNA in each of those two cancers. So in one cancer, there are something like 30,000 mutations, changes in DNA, and in the other, there are 25,000. And this is the complete list. So we now see uncovered all the forces that have generated that cancer, and we now see all the genes which are responsible for driving those two cancers.
So this is an important moment.
So this is a fundamental moment in cancer research. From here on in, we will think about cancers in a very different way. We'll think about them in terms of the numbers of abnormalities, the genes which are abnormal across the whole of the human genome, in individual cancers from individual patients.
And what does this mean ultimately for patients?
Now that we have these comprehensive, complete catalogues of mutations in individual cancers, we'll be able to see how each cancer developed, what were the exposures, what were the environmental factors, and that's going to be key for our understanding generally of how cancers develop. And for individual patients, we will see all the genes that are abnormal and are driving each cancer. And that's really critical, because that will tell us which drugs are likely to have an effect on that particular cancer and which drugs are not. And that will allow us to make much more informed choices in the future as to which drugs to treat patients with.